plant.
This meeting is being recorded.
So what will come first out of these four things? If I ask you root hair cells, xylem, root cortex cells, mesophyll cells. So where should the water start its journey from?
The root hair cells, miss.
Root hair cells. Okay, good. Second,
The root cortex cells.
third,
Mesophyll cells. Sorry, no. Xylem vessel and then the mesophyll cell.
yes, very good. This is the mistake that the students make. They first mention mesophyll cells. So it's first xylem and then mesophyll cells. Okay. This question will also test your concept. So state whether water is in the form of a liquid or a gas in each of these places. So yes, in A, is it a liquid or a gas?
As it moves from the soil into it's a liquid.
B,
As it moves to liquid.
and C?
As it moves out the gas, miss.
Yes, correct. Okay. And I hope you know the answer to this question as well. Explain the importance of the large surface area of the mesophyll cells in helping water to move through a plant. Yes.
Yeah, it just means that there are like more stomata present, which allow a greater rate of transpiration.
Correct. And also, it also means that, when we say the surface area of the mesophyll cells is large, so this means that it allows a lot of evaporation from the surface of the mesophyll cells into the substomatal air spaces and from there into the stomata. I hope you're getting what I'm saying.
I am.
Yes or no? Or should I show you the diagram? Very
Can you show me
quickly.
the diagram once again, please?
Because the diagram has to be seen. Look here. Now, this diagram shows you many spongy mesophyll cells. And look, if these spongy mesophyll cells, these are the ones, right? This one, this one, this one, this one. First of all, we've got lots of these, right? And each one of these has got a large surface area. And although it's not shown, but technically they should have shown a film of water. A film of water surrounds all these spongy mesophyll cells. So what happens is that the larger the surface area of the spongy mesophills, the higher is the probability that lots of water will evaporate from the surfaces of the spongy mesophyll cells. So lots of water evaporating means more transpiration and eventually more vapor in these spaces. Now this space is known as the substomatal airspace. It's better that you should know the name of this. So lots of water evaporating from the surface of the spongy mesophyll cells into this space, which is the substomatal airspace. And finally, where will this water go? Out of the stomata into the environment.
All right.
Right?
Okay, I got that.
You get that. Okay. Because these concepts, that's why I just wanted to do the questions with you so that I know. Or I think you know this as well, that if I ask you explain in your own words why water moves up through the xylem vessels in an unbroken column. So what things come to your mind when this question is asked?
Explain in your own words why water moves up through the xylem vessel in an unbroken column. This is because of that thing, right? The transpiration pull. That comes into my head.
Transpiration pull. But then, for example, this is the xylem vessel. And if I'm saying that the water is moving up in an unbroken column, it is not broken into pieces like these. So what are those properties of the xylem or the structural features of the xylem that enable it to do so? Unbroken column. The key word is unbroken.
So number one is like the lignin that is found that's really tough and rigid, meaning that it holds the water pressure together.
Correct.
When the water moves through it. And another one is that there's like a, what do you call, there's no there are no cross walls between the cells, meaning that the continuous flow of
Correct.
water that can pass through it is a free passageway.
Exactly.
I can't remember the other one, miss.
There was one thing which I explained to you, and what was that thing? I hope I have the diagram of that thing. Yes, this was the one. Remember this feature?
Yes. Wait, which one?
Adhesion and cohesion.
Adhesion and cohesion.
there was the fact that one water molecule is attracted to another water molecule, which is cohesion, and the fact that the water molecule is attracted to the wall of the xylem, which is adhesion. So these two sorts of attractions, they also play a role in pulling up the water in the xylem vessel in an unbroken manner. And these two forces together make up what is known as the capillary action. Remember, I told you about this.
Yeah, I do.
Right?
I just forgot the.
It's okay. So then this means that if you're asked about what are those features, or explain in your own words, how can water go up the xylem vessel in an unbroken manner? So you will mention about the transpiration pull, correct? You'll mention about the absence of cross walls, lignin, and also this thing should be mentioned. So it will be a comprehensive answer. Let's say, you never know, if the question is carrying 4 marks, 5 marks. So it will then satisfy all points. Now let's quickly move to this diagram without wasting time. Now, plants, they transpire, right? And when I say transpire, it means that there is a loss of water vapor. In fact, let me give you the definition, a proper definition of transpiration. Okay, so the definition of transpiration, I'm just writing it down over here so that you don't make any mistake, a technical mistake, while defining it. So transpiration is the loss of water vapor, and I'll be underlining the keywords as well. By means of diffusion. Achha. Now, what are the keywords, and what is the importance of using those keywords only and not any other words? Is you should not forget using the word water vapor for transpiration. Otherwise, if you simply write down water in a hurry, this mark is lost. Okay? This is keyword number 1. Second keyword is diffusion. The examiner should know that the process underlying transpiration is diffusion. Thirdly, this process is happening primarily, it can happen through any aerial part of the plant. Aerial means which is exposed to the air, to the sun, etc. But your answer will be incomplete if you don't mention the leaves. So usually this question carries 3 marks. Okay. And these 3 marks are attributed to these 3 key points which I have mentioned, right? So when you're done, let me know, and then I will move on to the next part.
This meeting is being recorded.
Okay, fine. Now, this is about it. Now, what happens is that one of the very important topics in transport in plants is to measure the transpiration rate. Now, since transpiration actually means the loss of water vapor, it is entirely impossible that we can get hold of the water vapor and sort of try to calculate the rate at which the water is being lost, right? Because everything is lost into the air. So what we do is in O levels also and in A levels, that we tell the students that the rate of transpiration can be measured indirectly. And that indirect measurement is that instead of directly measuring the rate of transpiration, we measure the rate of uptake of water by the plant, assuming that the rate of uptake of water by the plant is directly proportional to the rate of transpiration. Is that correct?
Yes, that's correct, Miss.
That's understandable, because the more the water takes up water, the more the plant takes up water, the more will be the rate of transpiration. So for that, we design an apparatus, and this apparatus, is taught in O levels, and then practically the students do it in A levels. So it's good that you should know how it works. So here you can see that there is a glass tubing. On the left hand side, the glass tubing is attached to a leafy plant. Achha, at every stage when I'm describing the apparatus, Hamza, you pay attention to what I'm saying, because all those points are the ones which the examiners ask. So the first thing that they ask, why did you choose a leafy plant? So then what will be your answer?
Why did I choose a leafy plant? This is because the surface area of the leafy plant is the highest, meaning that the transpiration will occur at the highest rate.
Correct. Okay, good. Now we move on. Why did you, okay, that I'll tell you afterwards. Then we want to attach the glass tubing to the leafy plant, and we have used a rubber tube with an airtight seal. So again, they asked, why the airtight seal? Because we don't want air to enter this water column and break the water column. So we are trying to simulate what is actually happening in the real world scenario inside the xylem. Are you getting
Could
it?
you repeat that, please?
You want me to repeat the point?
Yeah, at this point.
That point is, the examiner asks you, okay, let me just wait. The first thing was, why a leafy plant, right? And the second point is, why this rubber tubing? Okay, so the rubber tubing is used to seal this entire apparatus, because if you're not going to seal it, air will enter. Okay, and as soon as air enters, it's going to break the water column. There will be an air bubble. Okay, there will be an air bubble like this, and the air bubble will actually interrupt the water column. It will make it the water column should be ideally unbroken, right? But it will break it. So what happens is that the entire purpose of the experiment will be lost, because we, through this experiment, are trying to calculate the rate of transpiration indirectly by measuring the rate of water uptake. So we are actually simulating, okay, whatever is happening in the xylem. So for that, we need a continuous water tubing or a continuous water column. Acha. Then the third thing which you can see in this diagram is a water reservoir. This is very, very simple to understand. Why am I using a water reservoir? Because in the real world, what do? The plant is attached to the soil, and we are watering the soil, and we know the soil is wet, and there will be constant entry of water into the plant through the root hairs. But here we don't have any soil, so we have to have a constant water supply. So for the constant water supply, we have a water reservoir. Is that understandable?
Okay, so the soil isn't present, hence you need a constant water supply.
Huh. Achha. And then you can see that there is a screw clip over here. So it's just like a valve, okay? And this screw clip will be, if you want water to enter here, you will open this clip. If you feel that now this tubing is entirely full of water, there is no air bubble inside, you will close this clip, okay? So then this is very, very simple. Why the need for the screw?
Why the need for the screw?
To control, basically to control the water moving into and out. Okay. And then the last part, we have a capillary tube, a very thin capillary tube, which you can see. I'll just zoom into it. This one, it's so thin. See, this is thick, whereas this is thin. Okay. And they will again ask you, why are you using a capillary tube? So basically, we use a capillary tube because in this scenario, we will expect the water to be taken up by this plant. Okay? Just like any other plant, it's a living structure. So अगर हम इसको पानी में यहाँ डाल देते हैं, आहिस्ता आहिस्ता पानी will go, the water will go in this direction. Okay? It will keep going in this direction. The leaf will take up the water. When the leaf takes up the water, we know that the leaf takes up the water through the transpiration pull. So it's going to create a negative pressure in this part, a negative pressure, a suction pull. ठीक है, जैसे कि हमने straw से खींच लिया पानी. When that negative pressure is created, what happens is that this capillary tube, the level of the water in the capillary tube, it goes like this. For example, this is the start of my experiment. Okay, start में the level of water was somewhere here. अब अगर पानी ले रहा है plant and the water is going in this direction, what do you expect? Will this water meniscus start moving from this point to maybe this point and this point and this point and this point? Yes?
Yes, it will, miss. So the water meniscus will move inwards.
Okay, inwards. And then, although it's not shown over here, maybe it's written in the description. What do? We can use a dye or an ink to color this water, because since water is colorless, so it's very difficult to see through this glass tube. Okay, because this is made up of glass. This entire thing is made up of glass, and inside the glass, just as you must have seen a thermometer, na? So the clinical thermometer, which we use to measure the temperature when we are suffering from fever. So inside that thermometer, we have this mercury, which is shining, and that mercury rises or falls, which enables us to see. So that's why we will use a colored liquid over here, or we'll put some dye, red colored dye, so that we can actually see the water meniscus moving. So then the examiner asks you another question. Why should you use a colored droplet? Two things. Why a capillary tube? And why a colored droplet? So you will say, we will use a capillary tube so that we can see the movement of water. Since the capillary tube is thin, so the effects of negative suction pressure or the effects of pulling of water will be more obvious over here. And by putting a color inside this liquid, it will enable us to see this change easily. Right?
Okay.
Now, another thing is that, let's go back. We wanted to measure the rate of transpiration, and we wanted to have a mathematical value for it, right? In order to mathematically have a value for it, we are actually indirectly measuring the rate of water uptake. Now, the ideal thing should be that the water uptake could have been calculated in, ml. Okay. So the diameter of the capillary tube, it's a cylinder. So what's the formula for calculating the diameter or the radius of the capillary tube? Do you know that?
Of a capillary tube.
Because it's a cylinder shape.
Yeah, cylinder, it's gonna be 2 pi r height plus 2 sorry, 2 pi r height will be like the surface area of like the cylinder, right? And then for the top and bottom, it's going to be 4 pi r, I believe.
Correct. So do you think that by using all these mathematical formulae, we will be able to calculate the volume of the water?
Volume of the water, miss?
Volume of the water inside this capillary tube, for example, see, or simply, let's forget about the volume. I think it will confuse you. Basically, we can also do that let's say at the start of the experiment, the water meniscus was over here. Okay, over here. I'm using a black pen. It was over here at this point. After 4 hours, you keep this apparatus for 4 hours, the water meniscus was over here. So you measure the change in the distance, and the change in the distance will actually give you an idea about how much water was taken up during that time period. You can fix the time period. You can say, okay, I will see the water meniscus at time 0, and then I'll measure the water meniscus at the end of 2 hours or 1 hour, and I will see, I'll subtract them. Okay, I'll subtract the two readings, which will give me a value about the distance that the water traveled, the distance, okay, the water traveled in 1 hour. And that distance will help me calculate the rate of transpiration, because the assumption that I had made in the beginning was that the rate of water uptake is proportional to the rate of transpiration. Is that understandable?
Yes, it is.
Right? So you just measure the distance. Forget about the volume, because this volume thing will confuse you. And we do it usually in the A-level with A-level students, right? Acha. These were all the points. Wait. So I told you almost everything about it. And there's one more safety precaution which we use, and I did not mention over there, and that is that when we take a leafy shoot, okay, I'm on this point, just look at the screen. When we take a leafy shoot and insert it into the potometer, we take a slanting cut. For example, look here. This is the leafy shoot. Look at the way I'm drawing. If I have to, of course, I'm going to take it from a potted plant, okay, and I'll have to cut it. So what do? I make a slanted cut like this. By making a slanting cut and then pushing this shoot into the potometer apparatus, it will minimize the number of air bubbles entering, okay? And also that when I'm doing this cutting procedure, I have to do it underwater. Look here. I take a potted plant. I want to cut this much. Okay, itna mujhe kaatna hai. If I want to minimize the entry of air into this leafy shoot, what I can do is that I can do this cutting procedure under the surface of water. Maybe koi bada sa beaker leke, ek bada sa container leke, I put it inside, this whole thing inside the water, and then I take a knife and I give a slanted cut like this. Okay, aise karke. And then the examiner asks you in the exam that why did you do this? By doing this, you minimize the entry of air bubbles into this leafy shoot. Okay? In doing so, air bubbles nahi jayenge, aur jab air bubbles nahi jayenge, to jab aap isko finally attach karoge into the potometer, the air bubbles will also not enter there. Okay? I'm again reading this, the points 2 and 3. It says, cut the plant shoot, making a slanting cut, push the shoot into the potometer. If your potometer is like the one in figure 8.17, do this while the tube is still completely underwater. If you have submerged your potometer in water, take it out of the water and stand it on the bench. Check that your potometer is airtight and there are no bubbles. Is this point understandable?
No, not completely, miss.
I know majority of students don't understand it, but we, frankly speaking, you know, if you just remember, there's no need also to go into the depth. If you just remember, 2 points. Making the potometer airtight, number 1. Cutting the shoot with a slanting cut, okay? And pushing the shoot into the potometer and doing all this procedure while the potometer is underwater is going to minimize the entry of air bubbles into this apparatus. Bas, or kuch nahi hai iske. Okay? That is it.
Okay.
Achha. It says that repeat this investigation, but with the apparatus in a different situation. You can try each of these: blowing with a fan, putting it in a refrigerator. I'll explain what you mean by this. So basically, if you have a plant, okay, let me just draw a potted plant. And I'm drawing a plant which is growing under normal environmental conditions. And of course, we expect that transpiration will happen through the leaves of this plant. Okay. So Hamza, I am giving you 2 or 3 conditions, okay? And you will tell me what should happen to the rate of transpiration in these conditions. So condition number 1, high temperature. High temperature should increase the rate of transpiration or decrease it. Yes.
High temperature should increase the rate of transpiration, miss.
Okay, high temperature should increase the rate of transpiration. Answer is correct, but then tell me the reason why.
This is because you know how the water molecules are on the surface of the spongy mesophyll cells, right? And how they diffuse through the stomata outwards. If the temperature is higher, the rate of evaporation will be higher, meaning that more particles will leave the surface of the spongy mesophyll cells and hence diffuse through the stomata. And then obviously, yeah.
Correct. Very good. The only thing that was missing in your answer was, whenever you talk about high temperature, always talk about high kinetic energy and always make a link of kinetic energy with the rate of evaporation. Do you think high kinetic energy or high heat energy is going to increase the rate of evaporation of water?
Yes, it will.
Yes. So always mention, जब भी temperature high की बात होगी ना, whether you're talking about enzymes, whether you're talking about anything, ठीक है? High temperature का मतलब high heat energy, high kinetic energy, more movement of molecules. Condition number 2, increase wind speed. The speed of the wind is blowing very fast. So what effect will it have on the rate of transpiration?
Probably increase.
Okay, the answer is correct, but then think about the reason.
This is because the water droplets, or how they summon, or how they are collected on the lower surface area of the leaf. When the wind, obviously, when the speed of the wind increases, this means that I've learned this in physics. So more water droplets will be carried out, meaning that water droplets obviously won't be at a single place. So when this happens, basically, more water droplets will be replaced instead of those that are carrying it out. So it's basically like more space will be created, more or less.
Correct. Answer is 100% correct. Only one thing needed to be added is that when you say that the wind is blowing really fast, it is increasing the diffusion gradient for transpiration to occur. This is what the examiner wants you to write. Whatever you said actually can be explained in this one statement. It increases the diffusion gradient for transpiration. And what do I mean when I say this? See, when the wind is blowing, it's going to blow away all the water vapors which were lying here. है ना? Wind सारे water vapors को उड़ा के लेके चली जाएगी. When all the water vapors are blown away, this means that the diffusion gradient for the transpiration will increase. What do you mean by that? Leaves में ज़्यादा पानी हो जाएगा. Leaves will have a lot of water inside them as compared to the outside air. So hence, the ideal situation or the ideal scenario for increased transpiration will be set up. Is that clear?
Yes, it is, miss.
Okay, so you will use this word. Condition number 3. Condition number 3 is high humidity. What will it do to the rate of transpiration?
Humidity will probably increase, right?
No, humidity. High humidity means there are water vapors all around the leaf. But just like Karachi, have you seen Karachi's weather? Karachi is so humid because we live near the sea. So Karachi में बहुत चिपचिप वाली गर्मी होती है because बहुत humidity है.
Oh, so humidity means like the lack of drought, right? Or
yeah. Humidity means simply, पता है क्या? Humidity means lots of water vapor in the environment already. Lots of water vapor in the air. So do you think that if there is already lots of water vapor in the air, there will be a diffusion gradient for transpiration to happen?
And there's a lot of water. No.
So the rate of transpiration will be reduced. And the reason that you will say that the rate of transpiration is reduced because of reduction in the diffusion gradient. When you're done, let me know.
Okay.
Factors.
This meeting is being recorded.
Yes, by now understanding what all those factors which increase or decrease the rate of transpiration, we can summarize that dry, hot weather can actually result in a loss of excess water vapor through the plants. And if the weather is like that for a long period, it can lead to wilting of the plants like this. A plant which actually looks like this, healthy like this, because of excessively dry and hot weather, can wilt like this because of excessive water loss by transpiration, right? And wilting is a temporary phenomenon. if we re-water the plants or if we relocate it in a better environment, right? A temperature which might be a little cooler and environment which will be less dry. So the wilting can be reversed. But if it happens for a long time, the plant will die. So you should know this terminology of wilting, because I can just read over here that if the temperature is high, windy, and the air is very dry, then transpiration will happen very quickly. Plant may lose water from its leaves faster than it can take it up from the roots. So therefore, the individual cells in the plants, they lose so much water that they become flaccid. Okay? Cells become flaccid, but जो leaves में हमें नजर आ रहा होता है, we call it wilting. Many students, they ask me, Miss, what is the difference between flaccidity and wilting? Basically, they are both interrelated to each other. हम cells को कहते हैं कि they have become flaccid, but obviously the change that we see in the leaves, that when they curl backwards like this, we say it's wilting. Okay? So the tissues in the leaves are no longer supported by the turgid cells, pushing outwards against one another. The leaves become soft and floppy, and this floppiness and softness of the leaf is known as wilting. At a cellular level, they become flaccid. At a macro level, they become wilted. Is that clear?
Yes.
Right? Sure. I'll although
the macro level, the micro level, they become
at
the other way around.
Micro level, they become flaccid. At the macro level, they become wilted. Look here. If you see like this now, I can't say that the cells are flaccid. Can I see the cells in this diagram?
No.
No. So I can't say that this plant has become flaccid. It's wrong. I will say the leaves look wilted. The leaves look soft and floppy, right? Yeah, most probably this plant suffers water loss or water scarcity. And at a cellular level, the leaves, the cells might have become, the tissue cells might have become flaccid. Okay?
All right.
Right? Acha. The time is up, but then I will very quickly do one small topic so that next time when we meet, we don't have to, start, revise this chapter. We'll start from a new one. The only thing which is left is translocation of sucrose and amino acids. So I'll just teach it. I'll make a diagram and show you how this happens. So look here. Again, I'll be drawing a plant. These are all the roots. And inside the roots, I'm making some storage organs like potato. Potato is a storage organ. Okay, so now you tell me, where does photosynthesis take place in this diagram?
The leaf, miss.
The leaves. So where do you expect the glucose to be in the highest concentration?
The leaves.
The leaves. Okay, so we have lots of glucose over here. What is that? What is the name of the vessel inside the plant through which glucose and amino acids, not glucose, basically sucrose and amino acids are transported?
Sorry, miss, could you repeat your question, please?
What is the name of that vessel through which sucrose and amino acids are transported?
Phloem, miss.
Phloem. Acha. Did I tell you last time that why sucrose is transported and why glucose is not transported? Do you remember that?
I
think so.
Why sucrose is transported and not glucose? Yeah, that's perhaps I do. Is that because like sucrose can be broken down and then converted into glucose or fructose?
Glucose is more reactive, okay? And sucrose is not that reactive. That's why. That was the main reason. Okay, fine. Now just listen to me carefully and understand. Glucose is made, okay? And that's the end of photosynthesis. I would say that photosynthesis happens inside the leaves. Now I'm going to annotate this diagram. Photosynthesis happens in the leaves. And the end product is glucose. Now with this C, H, and O, molecule, the plant can do lots of things. Okay. The plant can convert this glucose into sucrose. It can convert it into amino acids. It can convert it into fats. I am not bothered about fats right now at all. I am only bothered about sucrose and amino acids. So let's start the journey. Glucose has been converted to sucrose and amino acids. And now these two together need to be transported from the leaves to all parts of the plant. Since their original site of production is the leaves, okay, and since they need to be moved not only to other leaves downwards, but to other flowers and fruits upwards, and to the roots downwards, everywhere inside the plant. So hence we say that the transport of sucrose and amino acids is bidirectional. It is not unidirectional like the transport of water and mineral salts. Okay, number one. So the first thing is that the transport of these two substances is bidirectional.
Okay. So it's bidirectional.
Bidirectional.
All right.
Another point, when these substances, since they are made inside the leaves and they need to be transported here and there and up and down. So in other words, we would say that they are translocated, isn't it? They are shifted from one location to another location. So again, when you are describing processes inside the phloem, the examiner wants you to use the terminology of translocation. तो इसका मतलब ये हुआ कि whenever in plants, I'm talking about plants only, you come across this word of translocation, the first thing that should come in your mind should be the transport of sucrose and amino acids in the phloem. So what do I mean when I say translocation? I mean this. इस terminology का मतलब ये है sucrose plus amino acids in the phloem. Translocation. Is this understandable up till now? If it is, then I'll move on to the actual part of it.
Yes, it is, miss.
Okay, now I'll start. And this part is a little confusing. Itna zyada nahi hai, you just need to pay attention. Okay? Acha, again, I'll repeat the same point, that translocation is the translocating or shifting of the location of sucrose and amino acids from leaves to other parts of the plant. Now, where can it possibly go? It can go from leaves to the stem. It can go to the buds, the young growing buds. It can go downwards to the roots. It can go to the storage organs. So in plants, while studying translocation, we have two regions, okay, two regions. And the name of those regions is sources and sinks. Sources and sinks. Okay? Sources are regions which lose sucrose and amino acids. Sinks are those regions which gain sucrose and amino acids. Okay? Sources are regions which lose sucrose and amino acids. Sinks are those regions which gain sucrose and amino acids. Now, let's start the journey of sucrose and amino acids over here. Let's suppose that this was your phloem, okay? This thing is your phloem. And inside the phloem, you have sucrose and amino acids. Sucrose and amino acids start their journey from the source over here, which is the leaf. So now I'm labeling this leaf as a source. And why am I labeling it as a source because it will eventually lose sucrose and amino acid. It will lose it. Why will it lose? Because जो भी यहाँ पे बना हुआ है अब ये यहाँ जा रहा है. It's going down and further down, and all this sucrose will be stored here. Sucrose stored here. In fact, stored as starch because the potatoes are rich in starch. Okay. So from this definition, from the point of view of this definition, the leaf was the source because it lost food. It lost sucrose and amino acids. The root, yahan pe, the storage organ root, becomes the sink because why? It gained sucrose and amino acid. So the region that loses sucrose and amino acid is the source, and the region that gains sucrose and amino acid is the sink. ये बात समझ आ रही है?
Yeah.
ठीक है?
I get that.
अच्छा, but always remember, now comes the confusing part. The students think the leaves will always be sources and the roots will always be sinks. The roles can be exactly reversed also depending upon the weather, depending upon whether it's a rainy season or it's a dry season. ठीक है? For example, in autumn when the leaves, when the tree is shedding leaves, they cease to become sources because they are being shed. तो अब कौन बनेगा source? ठीक है? Imagine a tree which is shedding its leaf in autumn और उसके पास बहुत छोटे-छोटे से नए leaves अभी थोड़ी देर बाद grow होंगे. So in that case, you know what will happen? During that particular temporary time, the leaves will no longer be sources. In fact, this responsibility will be taken over by the roots and by the storage organs. They will become sources. Why? Because the starch which was stored inside the roots, it can be broken down, okay, into sucrose and glucose, whatever, and then that sucrose can be transported from the roots to the young buds or the young tiny leaves which are now about to grow once the autumn season ends or starts to end. So depending upon the situation, the roles can be reversed. How will you determine that in exams? You will have to read the question properly. Okay? This has been mentioned. The example has been given in your textbook is that of the baobab trees in the wet season. जब बारिश होती है तो the leaves become the sources of sucrose. They are full of sucrose. They are sources. So all the sucrose will be translocated from the leaves to the stem and the roots and जो भी है. ठीक है? ये तो हो गया typical situation in the wet season. What happens when there is a dry season? जब सारे leaves खत्म हो गए, in the dry season, the stems and the roots, they store starch. उन्होंने पहले store की होगी starch. They are ready to supply the leaves with sucrose when they begin to grow in a few months' time. Got it?
No, could you repeat the last part again, Miss? So when they tend to dry up or?
When they, देखो, wet season, lots of water, normal situation, photosynthesis happening in the leaves, end product sucrose, amino acids, leaves will be the sources. They will provide sucrose and amino acids to all parts of the plant. ठीक है? अच्छा, अब बारिश खत्म हो गई है, and now we have a dry season. In dry season, the leaves, the tree has shed the leaves. अब जब shed ही कर लिए, तो how can the leaves be the sources? They can't be the suppliers of sucrose in this scenario. In this scenario, then the suppliers of sucrose and amino acids will be the stem and the roots. Basically, एक चीज याद रखनी है. Source means the supplier of sucrose. A source always loses sucrose and amino acids. A sink always gains sucrose and amino acids. So in this situation, in the dry situation, who's gaining sucrose and amino acids? The young leaves, जो छोटे-छोटे leaves अभी नए पैदा होंगे ना, they will gain sucrose and amino acids. And the stems and the roots will lose sucrose and amino acids because उनके पास इतना stored starch था that can be broken down, hydrolyzed to sucrose and given it to the young growing leaves जो अभी थोड़े दिन में पैदा होंगे. Now is this understandable?
Yes, that makes more sense. So it supplies. So during that time, so instead of supplying it, instead of the leaves supplying it, right?
Instead of leaf supplying
The
it.
roots and the stems supply it.
Yes. Right?
Right, that makes sense.
Huh. Okay. So, and another way to also remember it is that, you know, everything which is exam में ना ये use होता है कि anything, if you see something actively growing, new buds, new flowers, newly developed fruits, कभी इस तरह की कोई चीज हो ना, means के they are sinks. जो भी actively grow कर रही होती है ना, anything which is newly developed, tiny buds, tiny flower, tiny fruits, they are all sinks. They are sinks. Why? Because they need lots of food. And a thing which is fully developed, as a leaf is fully developed, so it has to be the source. This is also another way to find out what is the source and what is the sink. So whatever I explained you right now is mentioned on this page of your textbook. Go through this very carefully because जो आपका phloem वाला part है, it is only described on this page and on this page, and then your chapter is ending. So I would strongly advise you to thoroughly read this page. Inshallah, by today evening, I'll be sharing the notes of this chapter. So you can go through them. And do I take a test on Tuesday or from this chapter?
Please, by all means, miss.
Huh?
By all means.
By all means. Okay, good. So then be prepared for a quiz, and then my class will be a little longer because 30 minutes for quiz and then 1 hour for teaching. Okay?
Right, absolutely. Sure.
So we are done for today.